Connaissance, recherche, sciences,
mathématiques,
théorie,
physique,
philosophie, conférences, archéologie,
nature.
Okay. Good morning, everyone. So it's my pleasure to present to you some of our results on a broadly protective influenza vaccine. And also, I must say, I appreciate very much the open spirit of these lectures organized by Collège de France. So I'm very grateful to the organizers. Now, we have certainly tried to do something different from nature with our vaccine approach, but whether we do better, I think we should still be humble in that respect. So influenza, some other speakers have already touched upon the subject, but so it's well known it's caused by influenza A and B viruses. It causes typically annual epidemics. Direct medical costs are huge. There is excess mortality in each influenza season. And the main problem or the challenge to make a vaccine against this virus is that it's very diverse. So it has 16 hemagglutinin subtypes, which is one of the major glycoproteins on the virion. Also, the neuraminidase, the enzyme which is a target for Tamiflu, is a diverse, serologically diverse antigen. There's an animal reservoir. There are genetic reassortments, so there is a segmented genome, and as we've seen for rotaviruses, this can lead to exchange and selection of new reassorted viruses. And then there is the phenomenon of antigenic shift and drift. So if you look back over the past 100 years or so, the human population has been faced with a number of influenza A pandemics, such as the H1N1, the Spanish flu, Asian flu, Hong Kong, H3N2 variant, and then more recently, there is somewhat more activity apparently for the viruses in our human as a playground. So we have, of course, the bird flu problem, which is still not resolved, but mainly, of course, an agricultural problem. And then the Mexican flu, an H1N1, surprisingly an H1N1 variant, came up and probably will compete out the previously circulating H1N1 virus. So we are faced with a virus that is easily coping with our adaptive immune system, and making vaccines is not so easy for this virus. So the classical way of producing the vaccines is global monitoring. So WHO reference lab try to predict the virus which will circulate in the next season. Then there is a decision on which seed strain, so it means an H3N2 and H1N1, and then also a B virus strain, to be produced usually in embryonated eggs. More recently, there are some cell cultures available, but basically the vaccine manufacturers produce the virus, inactivate it. In many cases, it's split, so it's more purified than it used to be in the past. There are some attempts with fairly good success with a live attenuated vaccine, which is then administered as a spray. But eventually, the aim is to induce neutralizing antibodies, so antibodies that bind close or on the hemagglutinin near the receptor binding site, and then, let's say, prevent or try to prevent the infection. So aiming for a neutralizing vaccine response. But since this is highly variant, this antigen, this is not so easy to do for all the strains. And this is somehow illustrated here. So if you look at the seasonal influenza vaccines over the past years, you can see that the composition, so the seed strain that is used, is varying almost every year. And of course, last year we had the unexpected problem with the A. California H1N1, so the Mexican flu outbreak, which was named the pandemic. So this had to be inserted in this production system almost from the beginning. So in March, April, there was a need for this pandemic strain to be made as well. Now, the use of the influenza vaccines is also quite a difficult issue. Some people like it, some people don't. This is Belgium. It illustrates that in the northern part, Flemish-speaking people use more vaccine. This is for the Mexican flu than the Walloon or French-speaking people. Presumably, the Flemish people are more afraid, or were more afraid, or made more afraid by the media for the Mexican flu. So the use can be improved, and especially in certain target groups and in certain countries. This is an overview of the global capacity, more or less up to date. So it raised over the past 10 years. So we have now a capacity of about 800 million doses which is of course not enough for the global population, but it's improving. And also North America, or the North American countries are investing in new production facilities for the vaccine. So what would an ideal or a better vaccine against influenza look like? So first of all, one should probably target conserved viral strains or viral antigens, such that there is an extended protection. So you would probably need to induce both humoral and cellular immunity, to activate both arms of the immune system. The production system, as I've shown, is still rather old-fashioned, so it is room for improvement over there. And then such vaccines could have a number of advantages. Now, there are in the virus, in the viral antigens, let's say, there are conserved stretches, epitopes present even in the hemagglutinin, in the neuraminidase. Of course, the internal genes, the nucleoprotein M1, but also the polymerases are quite conserved, and these typically induce T-cell immunity. And we have looked mainly at the M2, so this is the third membrane protein of influenza, which has a very conserved ectodomain. Now, to go back a little bit to this T-cell responses, so this is an overview, a table with some CTL peptides from different proteins from the influenza virus, and you can see that they are fairly well conserved if one here compares typical human influenza virus with an H5N1 virus. So the same peptides usually come back also in birds, strains. There is some variation, but usually these are quite well conserved. Probably they protect us, and we find in many people responses against these epitopes, so we probably are all loaded with quite a strong T-cell armory against these conserved antigens, and presume they protect us they allow us to clear the virus, but not necessarily protect us from illness. This is just a small overview, but it's more elaborate lately, so people in the recent years have picked up quite a few B-cell clones, antibody-producing B-cells, which recognize on the hemagglutinin conserved parts. For example, this one is a single-chain antibody picked up by phase display, which recognizes a conserved stretch of the hemagglutinin. There's a trimer here in the stem region, which is conserved. There was also a study based on the fusion, or the part of the hemagglutinin which is conserved because it has a fusion activity, so it's has an important function in the infection cycle. Recently, from the group of Palese here, there is an antibody identified specific for H3 subtypes, which binds to this stretch of the hemagglutinin. So usually these antibodies are there, but they are targeting structures which are not necessarily well exposed, and it's certainly a difficulty. It's not easy to do the opposite and now try to raise antibodies which look like this by making the vaccine. So there's still a challenge over there. Now, our approach has been, as I mentioned, on the M2 protein. So just briefly, what is M2 doing? It's a proton channel, so it passively will open up its channel activity to let through protons, just based on an acidic environment or not. This is blockable by amantadine, a drug which is no longer useful now because most strains are amantadine resistant. This is a structure not so long ago published with the transmembrane domain of M2, so it makes 4 helices which are a little bit tilted, and the amantadine really fits well in the pore and presumably physically blocks proton entry. Not so long ago, there was a publication on that M2 also has an impact on autophagosome formation, at least the fusion of the autophagosomes with lysosomes, which is not seen in influenza B, and which is apparently also not proton dependent. The real physiological meaning of this in an animal model is not known yet. So a number of important functions for this small viral protein. So this is how it looks like schematically. It's a tetramer, has a very large cytoplasmic domain. Here we have the proton channel activity. There are a number of functions associated with the different domains. But what I will talk about mostly now is this conserved, this ectodomain, this small 23 amino acid tetrameric domain which is sitting out of the viral envelope and the infected cells. Now, it's not an obvious target to make a vaccine because if you look at the numbers, this is a cryo-EM from influenza virus, and you can actually see the spikes, the hemagglutinin and the neuraminidase spikes, and you can count them on the virus. M2 is simply not seen, it's too small. And if one does this a little bit more quantitatively, there are only a few, let's say 10 to 50 M2 molecules on the virus particle compared to the spikes which outnumber this protein. So it's small, and it's also not well presented in the virus, although it's essential to be there. but you can see it with the Western blot, for example. By contrast, on an infected cell, there's a lot of M2 expressed typically, which you can easily immunodecorate with an antibody. But so as a target, it is not very obvious. And most of all, there is no natural or no clear natural immunity, at least not B-cell immunity, in the human population. So after an infection, people will show clearly HI, hemagglutinin-specific titers, but low or non-existing M2 titers. So nature hasn't taught us to look for this target as a vaccine antigen. Now, what about the conservedness? Well, this is, of course, a major advantage potentially of this M2E. So this is a representation alignment of a few months ago of all human viruses and all avian influenza viruses. And you can see that at least the first 9 amino acids are in fact totally conserved. They don't change. Whereas in the second half or second two-thirds of the molecule, there are some variations, but even there are some conserved residues. And if we look carefully, for example, at this position, the glycine glutamic acid variation is the same as seen as in avian viruses. So the variation is there, but it seems to be quite restricted, and this has to do with the overlapping use of the genomic information for the M1 and at least the ectodomain of the M2 protein. So it's conserved, and in that sense, it's an interesting target to work with. Now, to make this antigen, this epitope domain immunogenic, we have used in the meantime, conventional approach. So we made the M2E fuse to the hepatitis B core antigen, which in E. coli can make virus-like particles. They are non-infectious, but they self-assemble fairly easily. You can easily purify them. And now, doing this, we got VLPs, which are fully decorated with M2E, and as such, could present this antigen fairly well to the immune system. We tried this in a mouse model, BALB-C, and we obtained high titer antibodies against the M2 peptide. We could show, importantly, complete survival after a lethal challenge, so controls die, M2E vaccinated mice survive. There is reduced morbidity. There is a reduction, we always see a clear reduction in virus lung titers. In protection, also an important aspect we noted was transferable by antibodies, so the immune serum, the antibody serum is really essential for the protection, or at least it's enough for the protection, but it works in concert with some other parts of the immune system. And then also intranasal administration of this vaccine candidate in the mouse model of protection. Now, since then, we have improved it because we wanted to move in collaboration with a company to a clinical trial. And first of all, we tried to extend to make the number of tandem copies of the M2E, so provide more M2E epitopes for the same amount of particles, let's say. And this improved the protection, so we had increased survival in the mouse model, lower lung virus titers, higher IgG1, IgG2 titers, compared to the, let's say, the prototype virus particle we produced in the past. Now, what about the heterosubtypic protection? So it's conserved, but then how does it do with different subtype virus challenges? So here, in this survival graph, I show you the vaccination of mice and then challenge with an H1N1 virus, so two kinds, one PR8WSN, an H3N2 virus, swine Belgium, which is a more recent isolate and actually has a quite different M2E sequence compared to the vaccine antigen. And then this is just for comparison, the situation with the M2 in H5N1 viruses. But it's clear that all mice that were vaccinated survived the challenge, whereas controls pass away. We did also an experiment with a kind of surrogate for the Mexican flu. So this is a PR8 virus which has the M2 from the Mexican influenza strain, so the 2009 H1N1 virus, and we have clear protection with our vaccine, also with PR8, and these are the controls. So in the meantime, we are doing the vaccination for a mouse-adapted Mexican influenza isolate from Belgium. So it protects apparently against many subtypes of viruses. And how can this be explained? Because I showed you the variation, and we have a single antigen to vaccinate with, a single M2E consensus sequence, let's say. Well, this is an ALASCAN, so we look at the expression of M2 FLAC variants in just transient transfection. This is the loading control. So every amino acid is mutated, except of course for the methionine. Then we look what the reactivity of this ALASCAN is towards 14C2. This is a monoclonal against M2E, and you can see that a number of residues are clearly important for this monoclonal to bind. So there's substitution in some of the the residues, there is no longer binding with 14C2. However, if we compare this with our M2E immune serum from mice, you can see that there is some reduction in binding, but still binding is seen at every ALA mutant. This is quantified over here by doing quantitative immunofluorescence and so comparing the flag versus the antibody signal. So the epitope, although it differs, the antigen in the vaccine seems to induce at least oligoclonal anti-M2E response, presumably allowing this broad protection in the mouse model. So then we switched to, we focused a little bit also on mucosal vaccination, which was done in the framework 6 collaboration together with a group in Göteborg. And this was, they have an interesting adjuvant which is based on cholera toxin. But instead of taking the B part of this cholera toxin adjuvant, they focused on the enzymatic part, so the ADP ribosylating function, which is actually the enzyme that kills the cells off eventually. And they fuse it to an immunoglobulin targeting domain. So as such, it does no longer bind to any cell in the body, because the B part is doing this. So now it's more restricted to presumably immune cells. And this adjuvant has been shown to be very safe, at least there's no toxicology, there's no toxic effect in rabbit models, for example. And we could see that intranasally administered M2E HB core particles combined with our vaccine did better than the vaccine alone. So this adjuvant will help our the protection in the mouse model. We took it then one step further. So we tried. This is a recombinant protein. So we inserted 1 or 3 M2E copies in between the CTA1 and the immunoglobulin targeting domain. So ending up with these 3 proteins. And again, immunized and challenged the mice. And as you can see, so this is the protection survival curve with showing as a control the M2E particles, so M2E VLPs with CTA1DD, so this is our positive control. Then if we use 3 M2E copies between the 2 domains of the adjuvant, we have full protection. If there's 1 copy, the antibodies are a little bit lower, and there's also still very significant protection. Lung virus titers go down in this model as well. So it seems to work combining the adjuvant and the M2E in 1 particle. We also, in that study, identified a T-cell response, class 2 restricted. Now, a second objective in our aim to improve the vaccine, because it's a small antigen, so anything you can do to improve is okay. So we made a soluble tetrameric form of M2E. So this is an NMR structure, at least a draft of it from the M2E fused to a GCN4 zipper, so it's a tetramerizing soluble, in the end it's a soluble protein, again produced in E. coli. This is the cross-linking study, and we also have mass spec data to show this. And this is an overlay of this structure, and then the consensus M2E waving conformation is here shown. But what is interesting is that the green helices over here, these are the same as the ones which were published in the recent NMR structure. So the orientation of these helices as they are in the M2 membrane protein very nicely fit with the orientation that the GCN4 zipper is somehow providing. So probably the way the M2E is here oriented to the outside is comparable to the M2E on the surface of M2 in infected cells, for example. This I will skip for the sake of time. So we showed protection with this M2E GCN4 and have some evidence for conformational epitopes. Now, a number of groups and many labs have tried to make other M2E fusions with different carriers. So VLPs have been tried, different ones than compared to HB core. Also other proteins or sometimes artificial oligomers of chemicals. And in many cases, or in all cases, when they did study with protection, other labs could confirm our findings, which is also reassuring that this is probably working at least in lab conditions. Now, the mechanism was an issue which is still not really resolved, and there are a number of studies in literature which are to some extent contradictory. So there was the group of Egerleiner claiming that it's really an NK-dependent mechanism of protection, but then this was not confirmed by others. There is a complement-dependent component, perhaps, based on some recent studies, and then there's even, based on in vitro study, potentially a direct effect on the virus. So it's not so clear how this vaccine actually works in vivo. And we decided to look at this by looking at a number of Fc receptor knockout mice. So we had access to the Fc common gamma chain knockout mice, which actually lack functionally then Fc gamma receptor 1, 3, and 4. And also we had a single and a double Fc gamma receptor 1 and 3 knockout to study. I will just show you one slide of this study because we did it quite extensively. So basically here we show that passive transfer of M2E serum protects mice, so the recipients are protected against challenge. If we do the same in the Fc common gamma chain knockout mice, there's much less or only residual protection. So clearly the Fc receptors are important. This is showing that the mice had the same antibody titers at the moment of challenge. This is morbidity. This is genotyping. We could show in this, we later show that it's important to have both Fc gamma receptor 1 and 3, and especially for IgG1 subtype antibodies in the mouse, the dependence on the Fc gamma receptor 3 Fc gamma receptor 3 is essential. And we also showed a role for alveolar macrophages by doing claudin depletion studies. But now I would like to shift towards the clinical trial that was on the phase 1 study. And we still thought there was room for improvement, so we had our original construct with 1 M2E copy. Now it was it had 3 tandem repeat copies of M2E. And we decided to compare the HB core carrier without or with a partially remaining nucleic acid binding domain. And this is just a Comassi gel to show these particles you can run on an agarose gel, and you can stain them with ethidium bromide, and then you can see that these 2 particles do bind RNA. This is RNA presumably from the E. coli, whereas the ones which lack this domain hardly do so. If you compare these 2 vaccines in the mouse model, we see protection with either of them, but the morbidity the morbidity profile here shown as the weight loss is much less pronounced. The mice are less sick when we use this vaccine antigen, and this was eventually then also put into the human trial. A second question that was raised was, what about your HB core? This is quite a few people will probably be HB core positive, and here we try to mimic in the mouse model the the presence of HB core, priming with HB core or PBS, and then follow with the vaccine. And we could show with 2 different adjuvants, hydrogel and MPL, that it didn't really matter if the mice were primed with HB core or not. So the response eventually to M2E was very comparable. So what about the phase 1 trial? So this product, 3 M2E copies fused to HB core with a partially full-length nucleic acid binding domain, was done in collaboration with a company called Acambis. They had a brand name for this, so it makes particles. There were some other studies necessary, like we showed protection in the ferret. Toxicology has been done, GMP materials produced, there was an FDA filing. So quite a lot of work to be done to go to the clinic. The production is fairly robust in E. coli, 30 liters. Theoretically, one obtains 80,000 doses if the doses we think of are protective. And there's also no animal-derived product, so there are a number of advantages for the production system. This is how the study looked like. So it's a placebo-controlled, double-blind, multi-center study with 4 arms, so 4 groups of 20 volunteers. There were no adjuvants used, or alhydrogel or QS21, a saponin-like adjuvant, together with either placebo, which is PBS, or the vaccine. There was an intramuscular injection at day 0 and 1 month later, and then serum was taken, blood was taken for analysis after that. And basically, the question in this phase 1 study was, is the vaccine immunogenic and is it safe? And both answers to the questions are yes, it's immunogenic, especially combined with QS21. So this is the seroconversion in the QS21 adjuvanted group. Alhydrogel didn't do much, and without adjuvant, doesn't appear to be much response in human. And also it's safe. There is some mild side reaction, but this is self-resolving. Now, what do these antibodies do? Well, of course, we want to know whether they are protecting humans, but for that answer, we can certainly not rely on these 20 people in that group. But we decided to look at the response of the human serum against M2, and here I show you a slide with M2 cells, M2 expressing cells, so these are the green cells. These are stained with human serum taken at day 0, so before the immunization. And this is from the same subject, the same individual, the day 60 serum, of course from a seroconverting person. And you can clearly see that the human serum binds M2 on the, in this case this is transfected cells. So the M2 that is raised by the vaccine does bind to the natural gene product from influenza. This is another study that tries somehow to bring the real life and the laboratory conditions together. So this is infection. This is adenoid-derived human tissue, which is then infected with, I believe, PR8 virus in this case. So, and it's known that the influenza will, at an apical, so in the polars, the cells of the epithelial layer, let's say, they have a polar orientation, so it's only at the site which faces the lumen, normally of the airway, that you have clear expression and budding of influenza. And we can see that over there we have nice staining with M2E immune serum from mice. So it's, and also we show it for different, infections with different viruses. So this brings me to the conclusions of the presentation. So first of all, M2 is highly conserved and as such is a very interesting target, but naturally it's not immunogenic, so we try to ignore nature's, what nature teaches us. The vaccination with these M2 VLPs protects mice against a lethal challenge for controls. The M2 VLP has been improved since our first study publication. Vaccination provides heterotypic protection. In this case, I mean heterotypic also for drifted or variant M2E carrying viruses. It's dependent on Fc receptors. The protection is enhanced by adjuvants, and some of them are acceptable for human. It's immunogenic in human, and it's well tolerated. The tetrameric M2E vaccine potentially could improve protection, but there we think we need a more robust or a multimeric form of the tetramer, so that's something we're working on. And then also mucosal delivery and protection through the mucosal route may be an interesting route to follow. So I'd like to thank the people in my group, and also I should acknowledge Walter Fiers, who is still very interested and helping in steering the project. We have a collaboration with Sanofi Pasteur, so Acambis has been taken over by Sanofi Pasteur a few years ago. We got some mice and reagents from collaborators. CT1DD was together with Göteborg, and then the recombinant viruses we can make because of the material we got from Eric Hoffman and Rob Webster. So I thank you very much for your attention.